Good afternoon, everyone. As we all know, Stargardt is the most common inherited juvenile macular degeneration, and ABCA4 gene mutation being the commonest in 85% of Stargardts. The ABCA4 gene is located in chromosome 1. Basically, it codes for a transport membrane protein on the membrane, which involved in all trans retinaldehyde removal for rhodopsin synthesis. So, whenever there is a mutation in ABCA4, there will be inefficient degradation, thereby excessive accumulation, apoptosis, and disease manifestation. Our purpose was to report novel phenotypic patterns, genotypes, and possible gene therapy prospects in Indian patients. It was an observational study with clearance from Institutional Ethical Board. This was the roadmap for our study to identify families with Stargardt disease, identify and isolate the mutation, studying these mutations in cell lines, and how to repair this mutation, and to study the effect of repair. Our study cohort had 39 families, 50 affected, 94 unaffected. For genetic testing, we used whole exome sequencing to identify and isolate the mutation. All patients underwent comprehensive evaluation and imaging. Electrophysiological tests were carried out with strict adherence to ICF protocol wherever it was required. So our cohort had 29 males and 21 females, age range between 9 to 64 years, and history of consanguinity was present in 28 families. That's approximately 73%. A spectrum of fundus abnormalities were observed. This was 28-year-old male with a typical Stargardt's with flex and atrophic macula, had homozygous mutation ABCA4. This was one more 30-year-old female with better vision, but there were no flex, just a ring of hyperautofluorescence. And there's one more 46-year-old patient with extensive degenerative changes with outer retinal cavitation and choroidal changes. All of them had a same mutation, but these kind of changes were never reported. So this was one more interesting family where the proband was 14-year-old female, mother was asymptomatic, the daughter had atrophic foveal changes, and she had compound heterozygous mutation in ABCA4. That means mutation at two different locations as opposed to homozygous mutation. But mother was homozygous for one of these mutation, but she was asymptomatic. When you did the autofluorescence, there were changes in the perifoveal region. A ring of hyperautofluorescence was seen. Maybe this is a very mild mutation which is causing mild disease in the mother. The combination of mutation is causing severe disease in daughter. This is typical example for a same family, same disease, same gene, but depending on the inheritance pattern and the type of mutation, the clinical features may differ. So this was a 43-year-old female. So it didn't appear like Stargardt, but we included in the study because father had a phenotype resembling the Stargardt's. So when we did a genetic sequencing, we ended up discovering a totally new gene which was pathogenic. But we are not sure whether this is responsible for this disease, but we are waiting for others also to report the similar conditions. Incidentally, the grandchildren were also carrying the same mutation, but they told they don't have problem. When we examined them, fundus appeared normal, OCT was relatively okay, but autofluorescence did had changes in the macula in the grandchildren's. This is the autofluorescence and the OCT image across 3 generations with increasing severity. So most of the mutation in our cohort had missense mutations. Missense mutation basically means there will be single base pair substitution leading to a totally new amino acids. Among these 8 were novel mutations. We wanted to study the effect of these mutations in cell lines. We did the same by studying the protein expression of ABCA4 gene using Western blot technique. To simplify things for the clinician, the darker the band, better is the protein production, like this VIEW mutation. Same gene, but these are different mutations. We went back and looked at the clinical photographs of this mutation. This is a 13-year-old male with the V type of mutation, had a very mild disease because of better protein production. Whereas second patient, 9-year, that is younger than the first patient, but had more severe disease because he had Q type of mutation. Even the ERG was also telling the same story. The first patient fulfilled was normal. The second patient, though the child is 9-year, he had severe rod cone dysfunction. So how to repair this mutation? So there is a challenge when it comes to gene therapy with a large gene like ABCA4. So we came up with a novel dual vector strategy where we split the gene into 2 of which were loaded into the separate viruses, and they were co-infected. Once they reach the target tissue, they combine back and where they are expected to function. These are the micrographs of our initial results in the mouse model. As you can see, as compared to the control, the dual vector take-up is very good. We use beta-galactosidase staining to trace this vector. That means the vectors are reaching where we intend them to reach. So to conclude, I would like to say what was known was Stargardt. This is how a Stargardt looks like, and most commonly caused by ABCA4 homozygous mutation. What this study adds is the genetic sequence and screening of Stargardt cases in Indian family, presence of new genetic and phenotypic variations, and experimental models for the pathogenicity of mutation. And we also had animal models to study the effect of dual vector. All these things, including the patient recruitment, vector production, enzyme, everything was done in India. So our ongoing projects, we have already produced a mouse model which mimics the human disease. This is one of the ABCA4 knockout gene. As compared to the wild type, there's increased autofluorescence. We injected dual vector and we did ERG. These are all alive mouse. We did ERG 6 weeks later. This was like a wow moment for us. As compared to the control vector and the non-injected vector, you can see the mouse which received dual vector injection had a better amplitude of A and B wave. Like someone was just asking me, like the newly approved Luxturna costs 6.5 crores. How are you going to bring down the cost so that Indian patients can afford? This is the way. So our future plan is to study the epigenetic factors, which is the limitation for this study, and also to start the human gene therapy trial. Thank you so much.
The vector that you injected into the mouse model,
Yes, sir.
what happened to the, how did you monitor the intraocular pressure? What was happening to the optic nerve of the mouse and all that? So how was the titration done for that?
Yes. So first when we inject, it is injected intravitreally. For example, if we inject 10 mouses, not 10 will go into subretinal space. Some will have, as you rightly mentioned, optic nerve damage, retinal extensive hemorrhage, vitreous hemorrhage, and cataract development. Post injection, we did a photograph of the fundus, the photograph and the ERG testing. Those who had a healthy bleb without all this complication which I just mentioned, only those mouses were included for the study.
So the ERG that you did for these mice,
Yes, sir.
um, was it not dependent on the injection also? Meaning not only the vector, but also on the trauma induced by the injection.
So this was a localized injection, sir. We used Hamilton needle, which is very small. This was a full-filled ERG. The damage which can happen because of the injection, which you rightly mentioned, which is a focal one. Maybe if you do a multifocal ERG over there, it may have changed. But what we did was a full field. We wanted to study the response in the entire, because the gene is mutation is present throughout the fundus. So that's why we did full field. Full field may not, it may negate the effect of the focal damage which can happen because of the injections.
Fantastic. Thank you. Thank you.
Thanks.